Hello everyone. Today in this video lecture we are going to study about carbohydrate metabolism. Now, what is this carbohydrate? It is a very essential nutrient required by every organism for the purpose of energy. Energy is contained in this nutrient that we call carbohydrate. As you eat rice, you eat chapati, pizza, burger, any kind of thing for the sake of energy. So what you find? You find carbohydrate. And in that carbohydrate, there are two chemicals or two forms, amylose and amylopectin. These are the forms which our body takes in the form of food, in the form of rice, chapati, anything. Now, what is the structure of this amylase? You can see amylase contains these monosaccharides. A single monosaccharide structure is like this. It's a 6 carbon compound, isn't it? So now our body cannot take this amylase. It cannot absorb this amylase. So what it takes? It takes glucose. What it takes? It takes glucose, the single monosaccharide, or fructose or galactose. Now the process in which this amylose and amylopectin are changed into the glucose so that it can be absorbed, that particular thing is known as carbohydrate metabolism. And this thing we are going to discuss in this video lecture. So as you can see, one monosaccharide is joined with another monosaccharide by this 1 and 4 glycosidic linkage. This is the first carbon, second, third and fourth. So one monosaccharide is joined to the other monosaccharide by 1, 4 glycosidic linkage. In amylose you find a straight chain and there is only 1,4 glycosidic linkage. But in case of amylopectin is branched. It has 1,4 glycosidic linkage as well as 1,6 glycosidic linkage. So there are two glycosidic linkages amylopectin which are needed to be broken down so that glucose would be formed and it can be easily absorbed. The main motive of the digestive system or carbohydrate metabolism is to grab this single monosaccharide, is to take this single monosaccharide from these complex structures so that it can be easily put into the blood and it should be easily used by the cells of whole body. Now what happens in the body? As you know, there are salivary glands present in the buccal cavity. These salivary glands release an enzyme and that enzyme is known as salivary alpha amylase. What this salivary alpha amylase do? It breaks this amylose and amylopectin into maltose, which is a disaccharide, maltotriose, you can see it looks like this, and alpha limit 1 dextrins. It has branchings also. Now you would ask, there should be a question in your mind, why this salivary amylase is not able to convert this amylose and amylopectin directly into glucose? Because this maltose, maltotriose and alpha limit 1 dextrins, they would not be used up by the body cells. Body cell only use monosaccharides. Then why salivary amylase is converting them like maltose, maltotriose or alpha limit 1 dextrins? Because salivary amylase can only act on this 1,4 linkages. Moreover, it acts only on 10 to 15% of food, because this is the first step of digestion which starts in the mouth, isn't it? So you got it. There are two reasons for it. First is very basic reason that only 10 to 15% of the food is acted by this salivary amylase and it breaks the amylose and amylopectin into maltose, maltotriose and alpha limit 1 dextrins. Now what happens then? In stomach we don't have a carbohydrate enzyme, but in case of small intestine we have pancreatic amylase. Now the 90% or 85% remaining carbohydrate needed to be changed into maltose, maltotriose and alpha limit 1 dextrins. This pancreatic amylase also can work on 1,4 glycosidic linkage and it breaks the amylose and amylopectin in same maltose, maltotriose and alpha limit 1 dextrins. Now in small intestine we have left with these things like lactose also, maltose, maltotriose, alpha limit 1 dextrins, lactose, sucrose, these things. which are not monosaccharides, which are disaccharides or oligosaccharides, isn't it? Now these disaccharides and oligosaccharides gets changed into glucose in small intestine, and for that we have brush border chemicals which are also known as succus entericus. What we have in our small intestine? Succus entericus. Now what is the constituent of this succus entericus? We have lactase, sucrase, maltase, isomaltase. These are the part of succus entericus which acts on these disaccharides and oligosaccharides. Now see, if we are having lactose is acted by lactase and it is changed into glucose plus galactose. If we have sucrose, it gets changed into glucose plus fructose. If we are having maltose, it gets changed into two molecules of glucose acted upon by maltase, isn't it? Now, if we are having alpha limit 1 dextrin, whom will act on it? This isomaltase, which will change it into glucose. Remember, isomaltase is the only enzyme present in the succus entericus which can cut this 1,6 linkage to change this amylopectin into monosaccharides. Got it? So now it is clear that we made the glucose in the small intestine. So this was the procedure by which we made the glucose. Now we have glucose or galactose or fructose. This is the lumen of the small intestine. These are the enterocytes of the small intestine, isn't it? And this is blood vessel. Let it be blood vessel where they are needed to be transported. There is a special protein known as GLUT4. This GLUT4 directly take this fructose and just get it into the enterocyte cell. This is facilitated diffusion. So for fructose we have facilitated diffusion via a carrier protein known as GLUT4. But for this glucose and galactose we are having a symport system. Which system? Symport system. What is this symport system? See, in each and every cell of the body we have sodium potassium pump. What is the sodium potassium pump? It takes 3 sodium out of the cell and 2 potassium inside of the cell, due to which sodium inside of the cell decreases. Same thing happens in the enterocyte. Enterocytes take sodium, these enterocytes take sodium out and potassium inside the cell, due to which what happens in this particular area, sodium gets more in the outside region and sodium becomes less in the inside. Okay, due to which sodium needs to be transported inside enterocytes. Okay, when sodium is transported in the enterocyte, what happens? It takes glucose and galactose with it. So now in enterocytes we are having glucose as well as galactose. Finally there is another carrier protein known as GLUT2 which takes this glucose, galactose, fructose directly to the bloodstream. So this was all about the carbohydrate metabolism. This was a beautiful concept which I taught you in this video lecture. I think you would be having confusion in one thing, this sodium potassium thing. So let me draw it again for you. See, in the lumen we are having more sodium, isn't it? But what happens inside enterocyte? Sodium quantity decreases. How the sodium quantity decreases inside the cell? See, there are certain sodium potassium channels which take 3 sodium out and 2 potassium inside the cell, due to which sodium inside the enterocyte decreases. So due to this gradient, the sodium transported to the enterocyte from lumen of small intestine, and this sodium is followed by glucose and galactose also, which is a symport type of system, and it is also known as sodium glucose transport, SGLT system.